So welcome everyone to the session Drug Development in Myotonic Dystrophy. And I'm actually not joking. We can talk about hope all we want, right? But in the end, we need to really measure it. And an indication of how successful we are as a community is if we look what's going on in the drug development community. So that's what we're going to do. A little bit of housekeeping. I provide a brief introduction. Then we hear from live representatives from companies, and then we have video presentations. I'll watch it like a hawk, 10 minutes, that's it, because we want to have, everyone should have a fair chance providing their background and their latest and greatest news. Okay. So drug development in myotonic dystrophy. You learn about that and you learn about soccer because it's very similar. When we look at here, we see a picture of Harry Kane. He's at the moment a forward for Bayern Munich, my favorite German soccer team. He was also the top scorer of the Bundesliga. And what you see there in these circles, that's positions from where he took shots on goal. And the lesson number one, he shot from many different positions. When we overlay that and look from which positions did goals occur, right? They didn't occur all from the same position. So the lesson for drug development is we need to take shots from different positions, and we need to take very frequent shots. Only this way we have a probability of making a goal. So the drug development process, like soccer, is a very strategic endeavor. And we heard this morning from Dr. Wang, right? The drug development process can be divided into very distinct phases. At the beginning, we have a discovery process that entails preclinical work, where we have basic research, we have early discovery, and then we have the preclinical work that will ultimately result in an IND, an investigational new drug application. When we get approval for that, then the drug candidate can move into the clinical testing phase. And as you heard this morning, right, the clinical testing phase is again subdivided into 3 phases: a first phase, a second phase, and a third phase, with increasing number of participants. The clinical phase ends with an NDA application, a new drug application, right? If approval is granted there in a review process by the FDA, we then go into the marketing phase, and in a phase 4 where we monitor post-market approval. Here is the big indicator of hope. This is a summary you can find on our website, myotonic.org pipeline. Here you see all commercial drug development programs currently underway in DM. You see that we have 9 programs that are in the clinical phase, and then many programs that are in the preclinical phase. And the really exciting news here is that while we have very strong leads from some of the what I call early adopters, right? We have a magnitude of programs. They have really interesting programs that are just sitting in the queue and are waiting to be executed and entering clinical testing phases. So clinical trial activities can be categorized into 4 broad areas, and they follow the basic dogma of biology, and this is shown here. So from left to right, we have the DNA with our genes and our repeats. Then through a process of translation, we form RNA. If we have the toxic repeats, right, of course we get misfolding, and these formation of these toxic hairpin loops. They act as very sophisticated traps for important cellular components. They capture those, and then they mess up important cellular functions further downstream. So the first category of drug development activities falls in this inhibition of transcription, gene editing, or gene tuning. We currently don't have anything in clinical trials here. The second broad category, that's where we see most activity, is the inactivation of toxic RNA. The third category is we have then compensation approaches for these trapped proteins, where companies go in and either add them back in or inhibit. And the last one is interfering with the furthest downstream mechanism, where programs aim to correct aberrant cellular function as a consequence of these trapped proteins. Here in these green boxes, right, that's where we have trial activity today. So we in this category 2, we hear from Avidity, from Dyne and PepGen. Compensation for trapped proteins is done by Arthex and AMO, and then compensation mechanisms happen by Juvena. Okay, so you probably get tired of hearing this. We have many programs, right? They all need to do clinical trials. So guess what I'm going to say? We need organization of the community, and we need you all to think about whether you would be willing to participate in the registry, because without a sufficiently large and well-characterized patient pool, we cannot do these clinical trials. Okay, so without further ado, welcome Avidity, and 10 minutes.
Yes, I got it. So you've heard the warning. Otherwise I would sit here and smile at you and get to know you for 3 minutes, but we got to go. So my name's Catherine Woods. I'm a medical science liaison, and I've been very privileged to be here today to share an update of our long-term data and discussion of the upcoming Harbor study. We've included a slide here. These are forward-looking statements. We are a publicly traded company. So these are educated guesses based on what we know now, and they may not be sure things for the future.
The notes.
Oh, they weren't there. Okay, no, that's okay. No problem. And so we, Avidity, we are committed to improving the lives of people impacted by diseases, including myotonic dystrophy, through the development of new drugs. Here, we're going to announce our new name for our investigational drug. If you followed the, I guess, the website or the press releases, you've seen the former name is AOC 1001. We were assigned a generic name, in which we've now shortened for easier communication. And that's how we'll be referring to it throughout this conversation, is Del-desiran. And here you can see a pictorial image of our antibody oligonucleotide conjugate in a picture form or an animation form. Here it kind of goes over our overview of study progression. And as the doctors have all talked about today, the importance of registry information and natural history studies. Without these, we would not be able to move forward with our programs as we have at this rate and with this amount of information and partnership with the community. So we do want to thank each one of you and your families, investigators, and the teams for your time, commitment, and continued contributions in our Marina and Marina OLE studies. So first we'll talk about the data surrounding our Marina OLE safety and tolerability. So in this study, the safety of the drug was the primary endpoint. If you remember, we did have a serious adverse event related. Let's see here. Sorry, well, hang on a second. Oh. Sorry, this is a little messed up here. So we did not have any serious adverse events related to the study drug. There was one in the Marina study. Avidity did take a look and took that very seriously and had a thorough investigation that was not found to be related to the study drug Del-desiran. All 37 patients enrolled in the Marina study continued on to the Marina open label extension. All adverse events related to the study drug were mild or moderate. None of the participants have stopped the study drug. And the most common drug-related side effects occurred were nausea and headache. Here you can see a pictorial diagram of the data regarding the natural and the NDM1 natural history study. You can see the gray plots on the line. The 0 is where you look at, sorry, did I mess something up? Stabilization. And as you can see here, there are several measures that we're looking at: video hand opening time, quantitative muscle testing, hand grip, and an activities of daily living measurement. And you can see if you move to the left, that's a declining score. And if you move to the right, an improvement score. So this is just from the NDM1 natural history. Here we will overlay the key endpoint using, overlay our Marina OLE versus our natural history. And so you can see here in the purple dots, these were patients treated with 4 milligram per kilogram every 13 weeks for 1 year. And you can see the purple dots now have moved and shifted over to the right of the 0, indicating, sorry, I don't know what this is, indicating an improvement in function. And now that I've showed you the data in a chart, here I would like to show you a video of one participant's experience in the Marina OLE study.
Ready? And squeeze, 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 and open.
So as you can see here on the left was the baseline hand opening time. And you can see on the right, this was a patient after 1 year of treatment in the 4 milligram per kilogram study group. And here we showed you in a video form, but here's the audio of somebody, one of our participants' testimony in the study.
I started this drug in June, and like 2 weeks after I took the first infusion, I went to open up a pop bottle, which I never would have been able to do. It was a twist pop bottle, and it opened right up. My strength was better. My outlook was better. My hands were working. That I had more strength and I could stretch them out. I could open things and I could turn doorknobs and all these things that were harder. Like my upper arm strength was better. I could walk better. I didn't need to wear my neck brace all the time. Everything just improved a lot.
And of note, this was one patient's experience. And so, others may not experience the same results, but we wanted to share some positive findings from that study. And here's probably what everybody wants to hear more about. As we advance hope, we're launching the next phase of our journey into this pivotal point in time and moving on to our phase 3 study, which we have named Harbor, is planning to initiate soon, as you see the dates listed there. Harbor being a global phase 3 study, as I said, the estimated start date is occurring currently. We are estimating to enroll approximately 150 patients with an age range of 16 plus. There's going to be around 40 global study sites. And this is thanks for the introduction to clinical trials, all the presentations we've had today. I don't have to go into it too much, but this will be a randomized placebo-controlled trial. And as you can see here, there'll be a study group receiving del-desiran 4 milligram per kilogram every 8 weeks and a placebo arm. And throughout that, there'll be a controlled study for 54 weeks. And then there will be an opportunity to enroll an open label extension trial as similar with the Marina and the Marina OLE study setup. And how to stay connected. You can visit clinicaltrials.gov for additional information on the Harbor study. You can search by, I think we've kind of talked about that today, search by disease state or keywords. But if you want to note down, that is the study ID for the Harbor study. You can type that in the search bar. You can please engage with the Avidity team members. We're here today. You've probably seen us around. And also, again, another plug to stay connected to the MDF registry is very important. And continue the consult with your doctor for questions about treatment options. And once again, we have a sincere and heartfelt thank you to each of you for your time, commitment, and continued contributions as we advance the world's understanding of DM1 together. And as I end, here's a picture of the team at Avidity that's working on del-desiran in partnership with you. Thank you.
All right. Next up is Dyne Therapeutics. Just going to start my timer here. So I'd just like to introduce myself. My name is Brian Newkirk. I'm the program lead for our DM1 program at Dyne. Dyne is developing several drugs, as you'll see in a moment, but our DM1 program is one of our two lead drugs. As with Avidity, please do note that, this is all, there may be speculation in here. If there's one thing I've learned in my, over a decade in drug development, it's that nothing is guaranteed. That is for sure. Dyne's mission is this. It is to develop life-transforming therapies for people living with serious muscle diseases. We were founded with DM1 in mind. We were founded with Duchenne muscular dystrophy in mind, and FSHD. And the tool we use to attack these diseases really is here. So this is how our drugs are put together. There are 3 parts. There's an antibody. It's actually not a whole antibody. It's just a portion of an antibody. I think my cursor is not showing on the screen, but I can, maybe this will, there we go, I think. So there's a portion of an antibody which targets a receptor that's on a lot of our cells called TFR1, and that takes the drug and moves it into the cell. There is a linker here that's actually in common use with other drugs. So this linker is in approved drugs and that type of thing. And that links this antibody to what we call here a payload. This is a little strip of RNA whose job it is to go into the cell. In the case of DM1, it goes right into the nucleus and uses just regular machinery that's part of your cell already to chop up the, in this case, toxic DMPK RNA. And the result is hopefully the hypothesis is that will allow the cell to go back to normal and restore all of the splicing, which many of you may know is kind of at the root of of DM1, as we see here. So, one of the big challenge, the root challenge in DM1 is that you have this one defect in the DMPK gene with these repeats, and it binds a molecule or several molecules, but these muscle-blind molecules whose job it is to make sure that all of these other proteins you can see coded by these genes take the right form. And if they don't, then you get problems with these proteins in the body. And as a result, you get myotonia and muscle weakness, you get cardiac challenges, cognitive impairment, and other brain difficulties. And that's why we see such a broad array of symptoms with DM1, and why one patient's DM1 is definitely not necessarily like another patient's DM1. So again, our idea is that we can restore splicing, we can correct splicing, and as a result of doing that, stop hopefully or improve at least a lot of these downstream symptoms. So what I'm going to do is I'm going to take you through the current data from the ACHIEVE trial. We released these data about a week ago or so, maybe 10 days ago. They show you kind of a midstream view of how things are going in this first in-human study of this drug. The ACHIEVE study is laid out at a high level here. So we're studying adult patients living with DM1, specifically between the ages of 18 and 49. This is just the first trial, right? This won't be the population that we, sort of eventually, hopefully get approved in. This is a first in human study, so it's studying safety and tolerability, which is, I think, another word for safety. There are also additional things that we're measuring, right? Importantly, we're measuring multiple assessments of muscle strength and muscle function, as well as what we call patient-reported outcomes. So these surveys where patients can say, even if in a non-quantitative way, how they're feeling and how they're doing along different dimensions. This is a 3-year study overall, but it's important to note that there's a 6-month placebo portion, and then another 6-month open label portion where all patients are on the drug, and then patients continue on for 2 more years after that. But we can see the results before we go through the entire 3 years. So the design of the trial is here. So this is the study looking to understand what dose and what regimen, meaning how frequently we need to dose, would be most appropriate for patients, would be, the maximum, maximally effective and also still safe. So we have enrolled now cohorts from 1.8 milligrams per kilogram all the way up to 6.8 milligrams per kilogram. Some of these are measuring monthly dosing, and then at the higher doses, we're measuring dosing every other month. And we'll use these results to assess what what we call go forward dose will be. So what we think the right dose is. And if we don't see sort of as much effectiveness as we want to see in these, then we can continue trying higher doses until we get to one that we think is really going to knock it out of the park for patients. This is an intravenously administered drug. So you kind of get a line in your vein, and you drip it in over between sort of 30 and 90 minutes. And in this study, we're also conducting muscle biopsies. So these are small kind of needle biopsies that take a sample from your shin, the muscle in your shin, the TA, to look at what are we doing with splicing in the muscle. Are we actually correcting the underlying defect of the disease? So I'll talk first about the most important thing in this study, which is safety. To cut to the chase, what we are seeing right now is a drug that is so far safe and tolerable at these doses. There are always adverse events in clinical trials like this. They may or may not be related to the study drug. There were 4 serious adverse events in the trial, which were all deemed unrelated to the study drug. One had to do with a cardiac complication. We know that those are common in DM1. One patient had pneumonia, and 2 patients had blood clots in their lungs, which are tied to actually other risk factors that those patients had. For example, one of them went on birth control, which is a known risk factor. The most common sort of regular side effects that we've seen, again, are not necessarily associated with the drug. The first one of these is a cold, nasopharyngitis, procedural pain from the injection, diarrhea, back pain, etc. We have also seen, as other developers have, liver enzyme elevations in about 20% of these patients. Now, this is complicated because DM1 patients also tend to have elevated liver enzymes. And so we've been looking very closely at patients' histories to understand if we're seeing, whether there's sort of new liver dysfunction as a result of the drug or not. We are not seeing any actual impact on liver function. You look at something called bilirubin to understand. So we're quite confident that we're not doing damage, at least based on the data that we've seen so far. And then finally, because we use this mechanism where we use this TFR1 receptor, which is also involved in iron transport and things that affect blood, we are looking very closely at anemia. So does the patient have enough iron in their blood? And so far, we've seen no evidence of any difference in anemia or thrombocytopenia. So let's cut to the data. So what you see here are data from 3 dose levels, 1.8, 3.4, and 5.4 milligrams per kilogram. The 1.8 and 3.4 are dosed monthly, and the 5.4 is dosed every other month. But what you'll see here is that the data from the 5.4 actually also comes after 3 monthly doses, because we have this kind of loading period at the middle. So you can compare apples to apples here. So if we look at how much drug is getting into the muscle, what we see here is what we call a dose-dependent increase in muscle concentration. And then we see that translating into splicing, which is incredibly encouraging, all the way up to 27% splicing correction. The other thing to note here is, as we've increased in dose, more patients have responded. So only 3 of 5 patients at the lowest dose saw splicing correction, but at the next 2 higher doses, all of the patients showed some degree of splicing correction. Now, not all of the patients achieved 27%, right? Some were higher, some were lower. Again, these are small patient numbers, so it's hard to say definitively anything, but this was very encouraging. So the next question we asked was, okay, are we actually affecting function? And the answer, I'll say cautiously, the answer seems to be yes, which is terrific. So in January, some of you may have seen the data that we showed going out through this point, 6 months at our lowest dose level, where we saw for that myotonia measurement of about a 4-second improvement versus placebo. We saw that extend out to an additional half second at basically 1 year. Now, as we go up in dose, we've seen increase in that response at that highest dose. And interestingly, at the middle dose, at 3.4, that effect was a little bit less. But when you look at where the patients started, these patients started at a much lower baseline VHOT. So they actually responded about the same in terms of percentage change. They just were already only taking 6 seconds instead of 11 seconds to open their hand, which is why it's the difference in terms of number of seconds is different here. Finally, I'll just leave you with what we've seen outside of just myotonia. So these are just some examples from the MD High, which is an instrument that's one of these patient-reported instruments. And we were very, very encouraged to see at 6 months and then a year continued improvement and maintaining that improvement at our lowest dose, and then additionally at the 3.4 milligram per kilogram dose, not just impact on that MDTI total score, but also impact in upper extremity function, gastrointestinal function, and fatigue. And we saw consistent across the other 14 domains of this instrument. We did see sort of the same sort of story. So, we're incredibly excited. It's, of course, early days. Want to give a huge thanks to the DM1 community, particularly the patients who contributed to the study design here as we were determining exactly how we were going to study this drug. Looking forward very much to hopefully coming again with more encouraging data down the road and to getting this study into registration. So thank you.
Okay, Jen.
Hello, everybody. I'm going to have to move this down. I'm not quite as tall as it. All right. Thank you, everybody, for being here and for still being awake. I know it's a long day, but hopefully we can stick through the last couple of sessions. So my name is Jenny Shoskis, and I work at PepGen in the clinical development group. So today I'm going to be talking about the molecule that we are looking at right now in adults for DM1. So PepGen, we're a small biotech company based in Boston, and really our goal is we are committed to changing the lives of people with neuromuscular and neurological disorders, including DM1. And we do this by really closely partnering with our community boards. And so here, this is a couple of ways which we've engaged with the community to make sure the design of our trial is actually something that makes sense to people and is reasonable. So things like including perspectives about the eligibility criteria in the study, asking about the order of assessments, or making sure that the amount of time that you're in the clinic isn't a 12-hour visit and by the end you're completely fatigued, and that might not be normally how you do that assessment. Providing tools and reminders for coming to visit. So making sure we give, a binder to people of, okay, here's your next visit. This is what you should bring with you. Here's what to remember. Here's approximately how long it's going to take. And then the last thing is, as much as we like to have a site in every single city in the country, especially in these earlier stage studies, they're going to be spread out. So there's going to be some travel involved, but making sure we can reduce the burden to patients and their families for that travel. So what I'm going to talk about today is our Freedom DM1 study. So we went through the phases of different trials earlier today, and we are a phase 1 trial. So we're really focused on looking at the safety of a single IV dose of the drug, which is called PGN-EDO-DM1, in people with DM1 compared to a placebo. So the total size of the study is around 24 people, and we currently are open in the US, in Canada, and the UK. So you can see 24 people, that's divided into 3 different dose groups. And so there's 8 people in each group. And in every 8 people, it's about a 75% chance of being randomized to active drug and a 25% chance of being randomized to placebo. So 6 would be on drug and 2 on placebo. And what does the study actually entail? So first, all of these studies have a screening period to make sure you fit the criteria, that you are a good fit, and that you are able to participate. The next would be the treatment period, so receiving either the drug or the placebo, and then following up, so making sure that there are no other safety events, that we can see if the drug is getting to the places that it needs to be. So the types of assessments and things that might occur during the trial include blood tests, urine tests, really to look at the safety, asking people about if they've had any changes in their medical history, looking at different strength and motor activities, asking you to fill out some questionnaires about your DM1, and then as well as doing a needle biopsy, really to see, are we getting the drug into the muscle and are we having any effect on the main mechanism of the drug? So the inclusion criteria of this study, we're looking at people that are between the ages of 18 to 50, have a confirmed diagnosis of DM1. There has to be a certain amount of strength in both leg muscles, which is where we're taking the biopsies from to make sure that can be done safely. And you need to have some hand myotonia. In terms of who may not be excluded in this study, congenital DM1, if there's any significant other diseases that may interfere with the participation, and if you're on any drug specific for the treatment of myotonia, they would have to be stopped before. So we only have a short amount of time today, but I wanted to point you guys to the resources. So we do have a study website and we are on clinicaltrials.gov, and we love to hear from the community, either with our clinical trials at pepgen.com or with our general inbox. The last thing I'll say, I think I've talked with many of you so far today at the table, and a big question is, there's so many companies, what's the difference between you guys? And the way I like to think of it is there's 2 main differences. One is how you get into the cell, and the second is what you're doing. So we have a slightly different mechanism that we use, a peptide that's attached to the drug to get into the different cells. And we've shown we can get into the heart, into the skeletal muscle, and into the smooth muscles and into the GI as well. And then the second part is how we work. So as we know, DM1 is caused by that expansion. I know it's probably very small, but those little letters are all the CUGs, that all those repeats form those hairpin loops. And what that does is that they act like a sponge, and it sucks up all the muscle blind protein that is normally needed in the body to process different things. And when it's all sucked up, then you have misprocessing of proteins and RNA. So our approach is to bind to those really long repeats to compete for that binding spot and let that MBNL1 not be able to get sucked up, to be able to get released from the cell and to hopefully go on and have normal processing and potentially improved symptoms. And we don't have patient data yet because we are currently enrolling, but what we do have is looking at a mouse model of myotonia. And you can see on the left is untreated. So when you pinch the hind legs of the mouse, you can see there is myotonia. They're kind of splayed out behind the mouse for a couple seconds. And then on the right, there we go, is after a single dose. So 2 weeks after a single dose of the treatment, you can see that mouse is scuttling along as normal. There's no signs of that hind limb myotonia. So we're really hoping, both from our preclinical data in the mouse and some other models, that this will really translate to people, not only with myotonia, but with other symptoms as well. So just a final thank you very much to everyone in this room for coming and showing up here and for talking with us, to our advisors who have helped us to design this study, as well as the physicians and sites that we currently work with. And I'll leave up this last slide in terms of our website, how to contact us. And we do have a table outside, and we love to talk with people. So thank you very much.
Okay, so I think the next presentations are recorded presentations.
Hi, I'm Jeremy O'Connell, co-founder and chief scientific officer at Juvena Therapeutics. And I'm excited to share with you today some of the progress our team has made in developing a new investigational protein drug for the treatment of myotonic dystrophy. Some of the statements about that progress are forward-looking and should be interpreted as such. At Juvena Therapeutics, our mission is to rejuvenate lives, medicine for movement. And we're doing this by unlocking the therapeutic potential of the proteins the stem cells in our bodies make to coordinate the repair and health of tissues throughout the body. By developing a proprietary platform that allows us to map the contents of those stem cell signals to their regenerative properties in order to understand where the best therapeutic application of them lies. In doing so, we've generated a growing pipeline that addresses major unmet medical needs in metabolic and muscle health. I'm excited to share with you today some of the progress for our lead program, a drug for treating muscle weakness and wasting called JUV161 that we're advancing into first-in-human dosing later this year, aimed toward the treatment of myotonic dystrophy. We've developed this drug to be broadly available to tissues from a subcutaneous administration and have strong preclinical evidence that demonstrates safety and the reversibility of aspects of muscle wasting and weakness. In myotonic dystrophy type 1, an expanded CTG mutation in the DMPK gene disables the function of that gene and the protein it would normally produce, and instead produces toxic RNA hairpin. This RNA leads to stresses within the cells, particularly muscle cells, that respond with an inability to take up and utilize energy for muscle fiber formation and the generation of strength or endurance during normal properties. This is called a loss of cellular homeostasis, where cells have a reduced capacity to respond to insulin to take up energy from their environment and to use that to fuel the formation of new fibers and the generation of muscle strength and function. So that collectively, this presents in the clinic as muscle wasting and weakness, respiratory defects, and myotonia, symptoms of myotonic dystrophy. To combat these aspects of myotonic dystrophy, we've created JUV161, a signaling fusion protein. And I'm showing you here an illustration of that drug, which is a protein, here demonstrated as a model with atomic level resolution of the backbone of that protein threaded against one of its receptors. This protein is a combination of one of the common carrier proteins in human blood called albumin, transports nutrients, and one of the drivers of muscle fiber formation during embryogenesis called IGF-2. Together, they become like an insulin for muscle, restoring the ability of muscle cells to take up energy for their environment and gives them a signal to grow and proliferate that allows these muscle cells to survive, differentiate, and produce strength properly. We've developed this into a drug that can be subcutaneously administered from a simple injection as one would with insulin. Here's some of the data that gives us conviction that JUV161 will successfully translate into the treatment of myotonic dystrophy. When we take cells donated by persons with myotonic dystrophy and grow them in a dish, you can see an example of what that looks like in this image on the left. The blue dots each represent a cell, and as the cells mature, they begin to express some of the proteins necessary for muscle formation, highlighted in green. As the cells mature, they fuse together into new fibers, which are formed of many muscle precursor cells. You can see in this example, not many cells survive, and few of them manage to succeed in forming new muscle fibers. However, when treated with JUV161, far more cells survive and far more form new, mature, large muscle fibers. As we quantify these changes between untreated cells, or those that would be like a placebo control on the left, here shown as the red bars on the right, we see that treatment with JUV161, the blue bars, have significantly better survival and new fiber formation. Importantly, when we test in mouse models of myotonic dystrophy type 1, here we see the mice lose strength week over week, showing a decline in roughly 8% in muscle strength, measured as the grip strength potential in these mice by the third week. However, treatment with JUV161 not only halts the decline, it enhances the strength above the baseline level. Collectively, these data give us hope of reaching our goal to restore normal function to muscle cells in people afflicted with myotonic dystrophy. Administration with patients may lead to improvement in the ability of muscle cells to survive and form new muscle fibers. The metabolic and structural properties of those fibers formed, and resultingly, improvements in strength and endurance capable of being produced from these muscles. Collectively, we're optimistic that if such achievements can be realized, it will lead to an increase in the mobility of people with myotonic dystrophy, and thus an increased capacity for social participation and an improvement in the quality of life. We're excited to begin dosing first people with JUV161 later this year in a study that will explore the safety and tolerability of JUV161 in healthy volunteers. In addition, we'll understand how the drug distributes and is metabolized by the body, and how the body responds to it in various ways, and which markers we can use to track the progress and response during treatment with JUV161. Zooming out one level to the longer-term study, we will first attempt treatment with JUV161 in healthy volunteers, following treatment of JUV161 in healthy volunteers, where we understand safety, tolerability, pharmacokinetics, and markers of the body's response to treatment. We will then advance to treatment in myotonic dystrophy type 1, in initially a proof of concept and then a pivotal trial to assess the improvements in muscle mass and muscle function over the course of treatment. As these studies progress and we gain additional conviction on the application of JUV161 to improve these important aspects of muscle strength and function, we will begin exploring additional indications where JUV161 has the potential to help people, such as myotonic dystrophy type 2, SMA, or sarcopenia. In summary, we're excited about entering the clinic in 2024 with JUV161. It represents an orthogonal approach to the treatment of myotonic dystrophy 1. As a natural signaling molecule that can drive muscle cell differentiation, survival, and strength, developed into an easily administered subcutaneous injection, similar to the insulin of muscle. We have strong preclinical safety data that gives us a first look into the anticipated human exposures needed for efficacy, at which doses we see well-tolerated and rapid-acting effects. Finally, as we look forward toward these clinical trials, we are excited to continue interacting with the Myotonic Dystrophy Foundation community to understand how we can minimize patient burdens and design trials that are representative of the community as a whole. We've enjoyed many of our interactions in past conferences and meetups and appreciate all of the feedback we have garnered there and look forward to many of the discussions we'll have in the future. Thank you. And please follow our progress at juvenatherapeutics.com.
Hello, everyone, and thank you very much for having invited me here to speak to you about our very interesting compound ATX01. My name is Judy Walker. I'm the Chief Medical Officer at Arthex Biotech, and I'm going to tell you how we have developed ATX01 and how excited we are to be going into the clinic very soon in a trial to look at the ability of ATX01 to treat patients with DM1. This is our disclaimer. And just to tell you a little bit about Arthex Biotech, we are a very small company headquartered in Valencia, Spain. And we have been working tirelessly over the last 5 years to bring really exciting development to the clinic. ATX-01. ATX-01 is an oligonucleotide drug, similar to some other oligonucleotides that you're familiar with, but different in that what we target with our oligonucleotide is a microRNA called miR-23B, and I'll tell you a little bit about that, but we think this is going to be really exciting for patients with not only DM1, but also DM2. So I'm not going to rehash how myotonic dystrophy is caused, but just to give you an example here of DM1, which involves a triplet repeat expansion in the DMPK gene, and we know how that DMPK gene DNA gets transcribed into RNA and forms these hairpin loops, and we also know how these hairpin loops trap MBNL protein. What I do want to focus on is the pivotal role played by the MBNL protein. MBNL is a regulatory protein, meaning what it does is it makes sure that other genes and RNA are translated correctly into proteins. So if you think of the gene or the DNA as the overall instructions and the RNA as the precise recipe for making proteins, there are many steps and processes along the way to make sure that we make all the proteins that we need and that our bodies function normally. Now, when the recipe isn't followed correctly, for instance, because there isn't enough MBNL to regulate the processes, then problems go awry, and that's when you start to see symptoms of DM1 or of DM2. So it's really important to have enough MBNL to do its job. Now, in myotonic dystrophy, the MBNL gets trapped in these hairpin loops of RNA, but so that reduces the amount of MBNL protein available to do its job. But it's not just that, because that's part of the story. The other thing that we know is that MBNL production is reduced. So not only is less MBNL protein being produced by the body when patients have DM1 or DM2, but it's being trapped. So there's two things going on. Now, why is MBNL protein not being produced? Well, this is where miR-23B comes in, because miR-23B is an oligonucleotide that for some reason appears to block the production of MBNL. Now, normally we don't have high levels of miR-23B, but DM1 patients have very high levels. The higher the number of repeats, the higher the levels of miR-23B. And so you can see that that will block production of MBNL. So whatever MBNL is being produced, it is much less than normal, plus it's being trapped. The other thing we know is that the miR-23B has an effect on the trapping of the MBNL in the mRNA hairpins. So there's a double whammy for miR-23B. So we thought, let's try to block miR-23B and see what effect that has. And that's exactly what we did. ATX-01 is an oligonucleotide that blocks the bad oligonucleotide miR-23B. So what happens when you block miR-23B is that you then allow the MBNL production to increase back to normal levels, plus when you block miR-23B, it causes the hairpin loops that are trapping MBNL to release the MBNL. So you now have plenty of MBNL from release from the toxic DMPK RNA, and because you've increased production. And therefore, the MBNL can go back to doing its normal job, which is regulating the splicing of other genes and therefore relieving the symptoms. The other genes that are involved, of course, are things that create the symptoms in DM1. So some of the genes that are regulated by MBNL include the insulin receptor gene. So if that's not spliced correctly, then you'll get a bit of diabetes. If your chloride channel is not spliced correctly, you'll get myotonia, etc. And this is why there's so many different symptoms of DM1. So that's how the drug, the ATX-01 works. But do we have any proof of that? And to make a long story short, yes, we do. We have lots evidence, and I'm just showing you here, and I know this is a busy slide, but one line of evidence from our HSA-LR mouse model. This is a mouse model that is used commonly in DM1 research. And essentially what we did with these mice is that we gave them either a placebo or we gave them different doses of our drug. And we found in all cases that what our drug did is that it relieved the muscle weakness, the grip strength improved when we gave them drug. The more drug they got, the better their grip strength. We noticed an increase in MBNL protein, not only in the muscle, but also in other tissues we looked at, such as heart, diaphragm, and brain. And this is important because we need to increase MBNL in all these tissues because there are signs and symptoms in all these tissues in patients with DM1. And we also increased the splicing index. Again, the more we gave, the more effect we saw, the more benefit. The trouble is, though, is that what happens if you give too much? Is there a limit? Can you get side effects when you give too much? And this is always the difficulty with drugs, finding a dose that will relieve the symptoms but will not be toxic. And we did a full battery of toxicology tests, and we found that we had lots of room to increase the dose well beyond the levels that we showed efficacy in the animal before we got any types of side effects. So this is very promising for when we go into human studies. So based on all these encouraging data, we have decided to go into a first-in-human study in patients with DM1. Now, I mentioned earlier that this mechanism of action of our drug should apply to DM2, to congenital DM, but we are starting with adults with DM1. And we are doing a very standard study with a design known as a SAD-MAD, single ascending followed by multiple ascending dose. The first goal of this study is to make sure that the drug is safe to be administered to human beings. We are confident on the safety in animals, but we need to be very, very cautious and check and see if that's the case in humans as well. We are also going to look at what happens when we give the drug to the MBNL levels in the muscle, and we're going to look at the splicing index. And of course, we're going to measure the myotonia, the muscle strength. We're going to measure walking. We're going to measure various other functions of the patients who are participating to see if we see any improvement. The study will be enrolling its first patients in the coming months. Probably first patient will be in the United States, and we are looking to enroll patients in Florida, in Kansas, in Richmond, Virginia, hopefully some other sites. We are hoping to enroll patients in Canada, in London, maybe another site in the UK, in the north, and in Europe, in Spain, France, and Italy, Milan and Rome. So keep checking our website and clinicaltrials.gov where you'll be able to find a list of the sites that are participating as soon as they become active. Who can be in the trial? Well, we want people to have a diagnosis of DM1 with at least 150 CTG repeats between the ages of 18 and 64, able to walk and have myotonia. We want patients to be off of anti-myotonia medication while they're in the study, and they have to be willing to undergo muscle biopsies. We are also making sure that we don't, in this first study, that we don't have people with advanced cardiac disease. And what does it involve? Well, after a screening period, there will be biopsies being performed, baseline assessments, and then in the single ascending dose, which is part 1, patients will get 1 dose, and in the multiple ascending dose, part 2, patients will get 3 doses 2 weeks apart, and patients will be followed up for 12 weeks after their last dose, with the assessments I mentioned earlier being conducted. And this is what it looks like on a diagram. 3 injections in the SAD, 1 in the MAD, and biopsies prior to the first injection, and in the SAD at day 15 or day 29, and in the MAD at day 85 or 112. So to finalize, I just wanted to say that we are very excited to be going into humans with this very novel mechanism of action of our drug. And just to remind you, we focus on the all-important MBNL protein level, and we increase it by 2 means: by reducing the toxic DMPK and releasing the MBNL that's trapped in it, and by increasing independently the MBNL protein production. We also emphasize that our drug should work in DM2, in congenital DM as well, because the mechanism of action is valid in those conditions as well. And also that we should see effects not only in muscle, but also in the heart, diaphragm, central nervous system, other tissues that are affected by DM1. And on that note, I'd like to thank you for your attention, and we look very much forward to continuing to engage with you in the future.
Hello, everybody, and welcome to this update presentation from AMO Pharma, discussing the work that we've been doing with the molecule AMO-02 in myotonic dystrophy. I'm very grateful for your attendance today, and I'd like to extend my thanks to Tanya and the team at MDF for the opportunity to present. I'll start off with a very brief reminder. Don't worry, I'm not going to go through this slide in too much detail, but I just wanted to repeat what we've said previously. AMO-02 is an investigational medicine. It's not approved for any prescription use at the moment. It's presented as a strawberry flavored liquid, which is swallowed, taken by mouth. Once it's absorbed into the body, it then moves to muscle, to brain, to liver, to GI tract, throughout the body. And once it gets there, it does two things. It interacts with an enzyme in the body called GSK3 beta, which is important in the control of tissue function in myotonic dystrophy. And it also binds to the CUG repeat RNA that is responsible for causing myotonic dystrophy. And we've shown that in test tube systems, and also shown that once AMO-02 interacts with the RNA in tissue from people who have myotonic dystrophy, that RNA is degraded. So this is a molecule that you take by mouth. It gets to all organs of the body and potentially has the ability to help with dysregulation of function in any tissue of the body. We've looked at this molecule in two studies in individuals with childhood onset myotonic dystrophy. We did an initial pilot study in children and adolescents with congenital and childhood onset myotonic dystrophy. That was a small study that's been published, and it gave us some reasons to believe that it was worth looking further at the potential ability of AMO-02 to help in myotonic dystrophy. Because of that, we ran a further study, which completed recently in children aged 6 to 16. You can see here an average age of 11 with congenital myotonic dystrophy only. That involved just over 50 children. Half of them received AMO-02 and half received placebo for a period of 5 months. And once that study was finished, all of the individuals involved were entitled to opt to then move into a further study, which is still going, where everyone gets AMO-02, there's no placebo, and that gives the ability of individuals to access the medicine if they think they're receiving benefit for a longer term. And we'll discuss that data in a moment. Now, in regard to the study we've just finished, this is a summary of the overall safety. And the key messages here are that during 5 months of treatment in the study that we just ran, the overall rate of adverse events was pretty similar on placebo or AMO-02 tiaglusib, as it's sometimes known. We didn't have any treatment-related serious adverse events. We did see, it's a known effect of AMO-02 that up to 10% of individuals will show increases in certain markers related to the liver, ALT transaminases, for example, and we saw that. But at the same time, we see in placebo-treated individuals, about 30% of individuals have problems relating to the GI tract, things like diarrhea shown here. And the incidence of those problems went down during treatment with AMO-02, possibly relating a treatment effect. Now, during the study, we looked at various different attempted ways of measuring whether or not the drug is helping. Our primary and key secondary outcomes focused on rating instruments where clinicians and the caregivers tried to assign a severity of symptoms on a sort of 0 to 4 scale. Those measures didn't help us really examine what was going on too much because we had a very large placebo effect, which is common in studies of children with disorders that affect their development. So what we focused on in our analysis is looking at functional tests, either of muscle strength or the ability to walk, or computer-based testing of cognition, or measures that can be looked at in blood samples that reflect what's going on in tissues. And some of the data are shown here, where on the top row, again, I won't go through it in detail, but creatine phosphokinase is a marker in the blood that shows whether or not muscle tissue is being stressed in some way. And there's a significant benefit of being on AMO02 according to that measure. And that's paralleled by, on the top row, the middle and right-hand side data, showing that the ability of the children in the study to walk 10 meters, the time it takes them to walk 10 meters, was speeded up if they were on treatment, but not on placebo. And on the bottom row, on the left-hand side, there's a blood marker called neurofilament light chain, which reflects neurons, whether they're, again, in some way stressed. And you can see, again, AMO02 produced an improvement compared to placebo. And that, at the same time, if you look in the middle and right-hand side graphs, we can see that a P-body picture vocabulary test performed on the computer, which measures IQ, or is closely correlated with IQ, showed a very high percentage, 2/3 of individuals nearly, did much better in terms of their cognitive skills by the end of treatment if they were taking AMO02. And I'm not going to go through all of this data. We can always discuss it in detail another time if you'd like. These are all of the functional and objective measures except for the neurofilament light chain, which is recent data, whether looking at muscle or cognitive function or the ability of the children in this study to undertake real-world tasks. On 9 out of the 11 things that we measured, people did better if they were receiving AMO02. Now, I mentioned of the 50 individuals that were entitled, almost all of them transferred to a long-term extension study where they've been receiving treatment, some individuals approaching 3 years of treatment now. And over that very long period of treatment, again, AMO02 has been generally safe and well tolerated, and benefits that were seen in the first study seem to be maintained. As a consequence of gathering this data, we've discussed what we found with the FDA in the US and MHRA in the UK. We've explained what we thought was going on, and on the basis of this, reached agreement on design of a study to look at AMO02 compared to placebo in what's called a phase 3 study in adults with adult onset DM1. And we'll be making an announcement very shortly that study will be taking place in multiple countries across the world starting in 2024. And that is really our update for today. If you have any questions, I'm sure you can get in touch with us via our website. Otherwise, I'd like to thank everyone who took part in this study, particularly, of course, the families for whom, it's really difficult to be in a study like this, and they did incredibly well, and we're so grateful for their participation. And that's it. Thank you so much.
Okay. So maybe a big thank you, and especially big thank you to those folks who attended in person, because there is quite a difference. So thank you. And for patients and families, where the tables are. If you have more questions, contact with them. Thank you very much.